Hi everyone and welcome. Thanks for joining me. So today I want to share with you a couple of technologies that will help you to measure the interactions of your membrane proteins, and that's even in close to native conditions. Now, I'm guessing that many of you are interested in membrane proteins because of how important they are as therapeutic targets. So being able to study the interactions of your membrane proteins is obviously really crucial. And because the lipid membrane environment of your membrane protein can have such a big impact on its conformation and its function, ideally you probably also want to study it in as close to native conditions as possible. Now, as you may have experienced, this can still be really tricky with many biophysical techniques, and especially when it comes to surface-based techniques, it still can be difficult to immobilize the membrane protein and have it maintain its native conformation and function. While it's fairly routine now to immobilize detergent-solubilized membrane proteins, it still can be challenging when it comes to things like lipid nanodiscs or membrane extracts. Because of the low yield and the instability of membrane proteins outside of the membrane, it may not even be possible for you to extract and purify your membrane protein into detergent. So when it comes to studying membrane protein interactions in lysates or other membrane mimics like lipid nanodiscs or something like SMALPs, then your choice of techniques can be quite limited. Well, I've actually been through something similar myself because I did work with membrane proteins for over 7 years during my PhD and postdoc. And a big part of that was looking at the interactions of a membrane protein called the TSPO and trying to understand its role in neuroinflammation. And really the only technique that I had available to me at the time for measuring affinities was the traditional radioligand binding assays. And I did these using crude cell membranes and also with purified membrane protein reconstituted into liposomes. So if you've done any radioligand binding experiments, then you probably know that these aren't ideal either. They're pretty tedious and they do rely on the use and the availability of radiolabeled ligands. But since then, when I was doing that work, there have been some great new tools developed, and they make it a lot easier to study membrane protein interactions, especially in sample environments like membrane extracts and different model membranes. So, I'm excited to share a couple of those with you today. I'm going to show you 5 examples of how the Prometheus and the monolith instruments can help you to measure membrane protein interactions, and especially in close to native conditions. Now, since detergents are still the most commonly used membrane-like environment that you'll use to solubilize and study membrane proteins, I'm guessing that you're still also interested in ways to characterize membrane protein interactions in detergents. A fairly straightforward way that you may have used of screening for likely interactions with a membrane protein is to do a thermal shift assay, where you see whether the addition of a potential binding partner changes the thermal stability of the protein, that is the melting temperature generally. You may have used one of the dye-based methods to do this, but maybe had an issue with detergent compatibility or the lack of resolution to detect small shifts in melting temperature. So in this first example, you can see how the researchers have got around this issue and performed a label-free thermal shift assay with the Prometheus instrument. That's to screen for possible interactions with a transmembrane transporter in detergent. The Prometheus instrument, what it does is it measures the thermal unfolding of proteins using intrinsic tryptophan and tyrosine fluorescence. And it's compatible with any buffers or detergents. So it's really well suited to measuring the stability of membrane proteins and looking for thermal shifts with the addition of ligands. So on the left, you can see the unfolding curve of the APO transporter in black, and then you can see how the addition of 2 different prodrugs each increase the thermal stability of the transporter. So this indicates that there is, or could be some kind of interaction. And then on the right, you can see how each potential binder that they tested change the thermal stability of the transporter. So this shows then which ones were likely to bind. So using the Prometheus in this way, you can see how you can really easily do a thermal shift assay to look for potential binders to your membrane protein without needing to use any dyes. And this gives you a good yes-no indication of whether something interacts with your membrane protein. But then I'm sure eventually you also want to measure the affinity of that interaction. And this is exactly what these researchers have done. They then went on to quantify these interactions using the monolith instrument. And what the monolith instrument does is it measures binding affinities that's in solution by detecting changes in fluorescence of your target molecule in response to ligand binding. And that fluorescence can come from a dye attached to your target protein or from the protein's intrinsic fluorescence. In this case, they use the label-free monolith to measure binding using the intrinsic fluorescence of the transporter in DDM. Now, in the graph on the left, you can see the binding curve for the interaction of one of the tripeptides with the transporter, either alone or in complex with a nanobody. And then by doing binding measurements using various mutants of the transporter, they could map the key residues involved in the binding. So you can see that studying membrane protein interactions in detergent is pretty straightforward with the Prometheus and the monolith. Both methods, they use very little sample and they're compatible with any detergents, and you don't need to worry about immobilizing your membrane protein. Now, while detergents are still the most widely used membrane-like system used to study membrane proteins, we do know that they're not necessarily ideal for maintaining the native or stable conformation of your membrane protein. And as I said, because the lipids surrounding your membrane protein in the native lipid bilayer can have a big impact on the conformation and function of your protein, you probably ideally would like to study their interactions in a model membrane that's closer to the native membrane. And there are now various ways to reconstitute your membrane protein into different model lipid membrane systems, but you might still be limited with the options to measure the interactions in these sample environments, especially when it comes to surface-based techniques. So in the next couple of examples that I want to show you, you'll see how you could use the monolith to measure membrane protein interactions in different types of model lipid membranes, like lipid nanodiscs and sapphisan lipid nanoparticles. And that's without needing to worry about immobilization. Now, in this example, they purified a GPCR, one of the serotonin receptors in detergent, and then reconstituted it into lipid nanodiscs of different lipid compositions. They then used the monolith to measure the binding affinity of serotonin to the serotonin receptor in the different types of nanodiscs. And you can see from the binding curves that the lipid composition of the nanodiscs changes the interaction of the receptor with serotonin. And in some cases, there is no binding at all. So in this case, you can see how the monolith, it allows you to directly measure the impact of lipid membrane composition and fluidity on ligand binding to your membrane protein. In this second example, the researchers wanted to confirm conformational differences of another serotonin receptor, a ligand-gated ion channel, depending on whether it was in detergent or lipids. This time, the receptor was reconstituted into sapphisan lipid nanoparticles, or SALIPRO, which were composed of brain polar lipids. You can see in the graph on the left how they've used the Prometheus to show that the receptor is more stable in SALIPRO than in detergent. And they confirmed with cryo-EM that the conformation of the receptor is different in lipids than in detergents. Then the graph on the right shows how they've used the monolith to confirm that the receptor in SALIPRO model membranes still binds its serotonin. So you can see how both the Prometheus and the monolith can help you to understand the effect of lipids on the conformation and function of your membrane protein. And they offer you a way to characterize interactions in model membranes that are likely to be more physiologically relevant. Now, if you want to get even closer to studying your membrane protein in its natural environment, or you simply can't extract it into detergent, then you've probably also looked into using detergent-free extraction systems such as SMALPs and SALIPRO that can extract membrane proteins while leaving the surrounding lipids intact. If you're not familiar with these systems, they can act a bit like a cookie cutter to cut out portions of the membrane, as you can see in the diagram. And you can then isolate your membrane protein of interest using size exclusion and affinity chromatography. So in this final example, the researchers, they've used the SMA polymer for detergent-free extraction of a ligand-gated ion channel, and that's the glycine receptor, while preserving the native lipids surrounding it. And you can see in the graph on the right how they've used the monolith to confirm that the receptor still binds glycine when extracted into SMALPs. So here, the monolith allowed them to measure the binding affinities between the glycine receptor and glycine and taurine, but in a way that it's in solution, it's embedded in its native lipid bilayer, and with both sides of the receptor accessible. So you can see how the monolith now makes it much more accessible for you to study the interactions of your membrane proteins in model membrane systems like SMALPs that preserve the native lipids surrounding your protein. And ultimately, this should help to give you even better insights into the interactions of your membrane protein target in vivo. I also want to mention now that if you can't purify your membrane protein target and you want to study binding in lysates, then the monolith also offers a great alternative to something like a radioligand binding assay. So if your membrane protein target has been expressed with a GFP fusion or a his tag, then you can use the fluorescence from the GFP or a specific his tag dye to measure binding affinities directly in the lysate with the monolith. So if you want to discuss any of these assay setups further, then do feel free to reach out to us and we'd be happy to discuss your particular project with you. So before I wrap up today, I just want to give a brief recap of the two technologies that you've seen today. The Prometheus allows you to measure the thermal unfolding and aggregation of your membrane protein using intrinsic fluorescence, so you can easily perform a thermal shift assay. It only uses a small amount of sample, it can measure a wide concentration range, and you can measure in any buffers, detergents, or lipids, or anything that keeps your protein happy. The monolith allows you to characterize the interactions of your membrane proteins in close to native conditions, so in solution with no immobilization required. Now you can work in any buffers or sample environments that keep your protein happy, and this includes things like lipid nanodiscs and SMALPs. So I hope you've had a good taste today of how the Prometheus and the monolith can help you to characterize your membrane protein interactions, especially in tricky sample environments and near native conditions. So to help you get better insights into your membrane protein targets. Thanks for joining me, and be sure to reach out to us if you'd like to learn more. We have time for a few questions now, so please feel free to enter them into the Q&A box, and I will answer as many as I can now. And if we don't get to your question now, then we'll get back to you as soon as we can with an answer.